Hi everyone, thank you for joining this webinar today. This is the last one of the year that we're organising in the SIMSIC Scientific Webinar Series. As usual, the attendees are muted, but obviously we would appreciate questions to the presenter, and we'll have time at the end of this presentation to take those questions, but feel free at that time either to raise your hand or you can type your questions into the chat box, and I can either unmute you after that or read them out. If we move on to the next slide, Yinglang has agreed to talk to us today. He's a senior pharmacokineticist at the Division of Quantitative Methods and Modeling, Office of Research and Standards, OGD at FDA. And he's responsible for managing research projects supporting bioequivalence studies, addressing controlled correspondences and citizen petitions, and providing response to pre-ANDA meeting requests, amongst other things. Member of the local acting PBPK team and has previously researched into ocular modeling. But the focus of the talk today, and we're really happy that he agreed, is relating to a publication at the back end of last year. So we're keen and we're patient with you to come and give us your presentation. Thank you for taking the time. But the reason why we contacted him is because it was a really interesting publication linking the prediction of drug-drug interactions, not only at the level of enzymes, but also investigating the role of gastric pH-dependent DDIs, and obviously that links into FDA guidance on this topic as well. So I'll hand over to you, and thank you again for agreeing to talk to us today.
Thank you, and thank you very much, Oliver, for the kind introduction. Hello, everyone. Again, my name is Minliang Tan, and I'm a senior pharmacokineticist in the Division of Quantitative Method and Modeling, Office of Research and Standard within FDA, OGD. First, I really like to thank SIMSIP Scientific Webinar Organizer, Dr. Masoud Jami, for the invitation, Oliver and Gabi for the logistic help and support. It is really my great pleasure to share our recent research on using PBPK modeling to predict drug-drug interactions between omeprazole, and which is a proton pump inhibitor, and with extended release nifedipine with different formulations. So here a disclaimer. First, I would like to state that this presentation reflects the view of myself and it should not be construed to represent FDA's policy and or view. Yeah, as Oliver mentioned, today's presentation is related to our recent publication in CPT-PSP entitled Use of PBPK Modeling to Understand the Effect of Omeprazole Administration on the PK of Oral Extended Release Nifedipine. Again, thanks to SIMSIP team for giving me this opportunity to share our work. So today's focus will be on the applying a PBPK modeling approach to understand the mechanism of complex DDIs between omeprazole and the extended release nifedipine with different formulation designs Due to the time constraint, I may not be able to cover every detail of this study today, but you could refer to this application for more information if needed. First, I would like to give you some background information for this project related to gastric pH-dependent drug-drug interactions. Then, a brief introduction to our recent clinical studies that was related to this modeling research project, followed by PBPK modeling study that was intended to understand the complex DDI mechanism between nifedipine and omeprazole that was observed in that clinical study, which includes dissolution testing that was used to characterize the release profiles of different nifedipine ER formulations, which will then be utilized to build Nifedipine PBPK models. Nifedipine and omeprazole-based model building and validation, and the drug-drug interaction study using PBPK modeling approach to understand the mechanism, specifically to understand if the DDIs be mediated by gastric pH and or CYP enzymes. So finally, some take-home message. Why is it important to assess gastric pH-dependent DDIs? As you may know, elevation of gastric pH by acid-reducing agents, in short ARS, can affect solubility and the dissolution characteristics of some orally administered drugs. As a result, concomitant administration of a drug with an ARA could change the bioavailability of the drug, potentially resulting in a loss of drug efficacy or increase adverse event. Here is a slide that I borrowed from my colleague, Xining Yang, who presented in 2023 December SBI seminar, showing different ARAs such as atanside, for example, aluminum hydroxide, magnesium hydroxide, magnesium trisilicate, calcium carbonate, histamine H2 receptor antagonist, or H2 blockers such as formotidine, ranitidine, nizatidine, cimetidine, and proton pump inhibitors or PPIs such as omeprazole, esomeprazole, lansoprazole, dexlansoprazole, pantoprazole, etc. These ARAs are widely used, and some are available through prescription and also over the counters. So basically, the gastric pH may be elevated after orally taking these ARAs. That may affect the solubility and the dissolution of the oral product. Subsequently, alter the drug bioavailability for the orally administered drugs. And they consequently, for example, decrease drug efficacy, for example, for weight-based drugs, while increase adverse event for weak acid drugs. Therefore, there is a potential risk for clinically significant pH-dependent DDIs when taking a drug concomitantly with an ARA. These complex issues in gastric pH-dependent DDIs have recently prompted the US FDA to issue a federal registry request to collect public comments and input, and subsequently published guidance for industry entitled here, Evaluation of Gastric pH-dependent Drug Interactions with ARA study design, data analysis, and the clinical implication in March of last year. This guidance recommends the sponsors to assess the potential of pH-dependent DDIs. So most drugs that have demonstrated pH-dependent DDIs are weak-base drugs, which have low intrinsic aqueous solubility. If not completely dissolved in physiological pH for IR products, an increase in gastric pH would result in a decrease in dissolution and also subsequently reduce the drug absorption rate and or extent. So this guidance recommends a stepwise manner based on the physical-chemical properties of the drug substance and also dissolution profile of the drug product. For weakly acidic IR drug product, the drugs are not completely dissolved in gastric fluid at physiological pH. An increase in gastric pH may lead to an increased drug dissolution and subsequently drug absorption rate and or extent. For the weak acid IR product, there is a relatively less experience with the evaluation of the impact of pH-dependent DDIs as compared to weak-base drugs. Lastly, for modified release or MR products, there is a very limited experience with clinical pH-dependent DDI evaluation for these types of drug products as compared to IR product mentioned above. The current study belongs to this category, that is modified release product. So recently, we have performed clinical studies that was intended to study the potential effects of omeprazole medication on PK of nifedipine ER products with different formulation designs, where 2 nifedipine ER tablets products were tested with or without short-term omeprazole co-administration in healthy subjects. This was an open-label, randomized, single-dose, full-sequence, full-period, 4-week crossover phase 1 study involving nifedipine ER tablets with or without omeprazole capsule under fasting condition. Details about this clinical study can be found in a recent publication in CPT, so I will only give some highlights point in next couple of slides. Why this clinical study? As we just briefly mentioned, nifedipine also is a calcium IR influx inhibitor, and the nifedipine ER formulations are indicated for the treatment of angina and hypertension. As you may be aware of, for the ER tablets, there are both for osmotic pump and the matrix-based release formulation designs for the product on the market, which may have different drug release characteristics. For example, pH-independent release for osmotic pump formulations versus pH-dependent release formulations with matrix design for the formulation. On the other hand, the gastric pH may change under certain conditions, such as in patients who take PPI for the treatment of gastric acid-related disorders, as we just discussed earlier. So the question is, if the gastric change caused by the co-medication of proton pump inhibitor omeprazole affect the PK of nifedipine ER formulations? To answer that question, this clinical study was performed. Again, this was a open-label, randomized, single-dose, full sequence, full period, 4-week crossover phase 1 study, using nifedipine ER tablet with or without omeprazole capsules under fasting condition. So there are 4 study arms, arms A to D. Arms A and B used nifedipine ER tablet only, without omeprazole pretreatment, where arm A used nifedipine ER tablet that has pH-dependent dissolution rate, while arm B used osmotic pump-based nifedipine ER tablet that has pH-independent dissolution profile. I will show you the different pH-dependent versus pH-independent dissolution profile in a later slide. For arms C and D, both studies had 7 days of omeprazole pretreatment, followed by a single dose of nifedipine ER tablet, where arm C used the nifedipine ER tablet that has pH-dependent dissolution rate, while arm B used osmotic pump-based nifedipine ER tablet that has pH-independent dissolution rate. On the right panel here showing is the observed time plasma concentration time profiles in healthy subjects for 4 arms. Again, arm A and arm C showing in orange and red are the result using pH-dependent nifedipine ER formulations without versus with 7 days of omeprazole pretreatment, while arm C and arm D showing in black and blue are the result using pH-independent nifedipine ER formulation, that is osmotic pump design without versus with 7 days of omeprazole pretreatment. You may see different PK profile in different arms with different study design. Here is a summary of the PK parameters from this different study arm. On the left side of the table are the results from arm A and arm C using pH-dependent release nifedipine ER formulations without versus with 7 days of omeprazole pretreatment, while on the right side of the table are the results from arm D and arm B using pH-independent release ER nifedipine formulations with versus without 7 days of omeprazole pretreatment. Highlighted in green box are the AUC and the Cmax ratios between nifedipine ER formulations with versus without 7 days of omeprazole pretreatment. As you may see, omeprazole pretreatment increased the geometric mean ratio of AUC and Cmax to about 133% and 113% respectively for nifedipine ER formulations with pH-dependent release formulations, while for those with pH-independent release tablets, the AUC and the Cmax ratios were 121% and around 123% respectively. So the bottom part of the table was the statistical analysis for the subpopulations whose gastric pH was less or equal than 3 in arms A and B, and also pH was greater than or equal to 4 in arm C and D. So as you may see, similar increase in AUC and the Cmax ratios were observed for both pH-dependent and osmotic pump-based pH-independent nifedipine ER tablets products in this subpopulation. So overall, systemic exposure of nifedipine was increased in subjects after multi-dose pretreatment of omeprazole with both formulations. Here is the summary of the clinical study. So there was maybe clinically insignificant, but statistically significant increase in Cmax and AUC of oral nifedipine ER tablets after short-term administration of a therapeutic dose of omeprazole. Drug-drug interaction was observed between omeprazole and nifedipine ER tablets with different formulation design. However, a remaining question was if the observed DDI is mainly mediated by the elevated gastric pH or not, since the DDI mechanism has not been well understood, as the DDI may also be mediated through CYP3A4 enzyme inhibition, in addition to the elevated gastric pH caused by the co-administration of omeprazole, as shown on the next slide. So there are a couple of potential drug-drug interaction mechanisms between the nifedipine and the omeprazole. First, as we discussed earlier, the elevated gastric pH by pretreatment of omeprazole and proton pump inhibitor may affect the release of the nifedipine ER formulation and subsequently change the systemic exposure of the nifedipine. Secondly, the DDI may also be mediated through CYP enzymes, as omeprazole may be metabolized both by CYP2C19 and CYP3A4 enzymes. Omeprazole and its metabolites may also inhibit CYP2C19 and CYP3A4, and on the other hand, the nifedipine is also known to be a substrate of CYP3A4 and CYP3A5 enzymes. So the question was, if the omeprazole and the nifedipine DDI is mediated mainly by elevation of the gastric pH and or CYP enzymes. So to answer this question, we use the PBPK modeling and simulation approach to investigate the underlying mechanism of this complex DDIs between omeprazole and the nifedipine. So first, let's take a look at the omeprazole and its metabolites. Omeprazole formulation contains racemic mixture of S-omeprazole, in short S-MOMP, and R-omeprazole, in short R-MOMP. So in enantiomer specific metabolism is observed with omeprazole, as the two enantiomers are both metabolized by CYP2C19 and the 3A4 enzyme, and the contributions of this enzyme are analyzed in vitro and reported to be enantiomer specific, so which means with different rate for different enantiomer. Two of the main metabolites produced by CYP2C19 enzyme mediated clearance of omeprazole was 5-hydroxyomeprazole, in short OHMOP, and 5-O-bis-methylomeprazole, in short DMOMP. Based on the in vitro analysis, This metabolite seems to be important in the reversible and the irreversible inhibition of CYP enzymes. So we can tell from here, this is like a very complex time-dependent DDI here involved. Our initial Omeprazole PBPK base model was developed by my colleagues about 10 years ago, where stereoselective disposition of each enantiomer, R omeprazole and S omeprazole, were described in the model. So recently, this model was expanded to include major active metabolites, hydroxy omeprazole, and this methyl omeprazole, and also updated with the new in vitro and in vivo data available to the model. Showing here is a model structure describing the time course of both omeprazole enantiomer, S omeprazole, and R omeprazole, which is on the top, showing with light blue background, and the two of its main metabolites, DM omeprazole and OH omeprazole, which is on the bottom part of the figure, showing with yellow and purple background. The blue X symbols showing auto or mutual inhibition between both omeprazole enantiomers, while red X symbol showing the inhibition of both metabolites on S and R omeprazole enantiomer in the guts and the liver, where time-dependent inhibition of CYP2C19 and CYP3A4 enzyme. As you may see, the inhibition mechanism is complex. Shown here is the concentration time profile following a single dose of 20 milligram omeprazole in enteral-coated tablets in healthy subject. On the top right is for the omeprazole, and on the bottom of the figure is for its two metabolites, OH omeprazole and the DM omeprazole. Solid lines are the simulated result, while dots represent the observed data. As you may see, overall, the updated omeprazole-based model will capture the observed PK profile for omeprazole. Now, let's take a look at the Nifedipine PBPK base model. So, Plato et al. From SimSafe built a full PBPK distribution model about also 10 years ago to describe the Nifedipine ADME process after oral administration to humans. Using literature and their in-house data, showing on the left panel was their PBPK model structure. On the right side was the predicted Nifedipine plasma concentration profiles for IR and controlled release formulation compared with the corresponding experimental data. As you may see, the model predicted the observed data reasonably well under both fasted and fed conditions. Utilizing the omeprazole and the Nifedipine PBPK baseline models we just described above, now let's take a look at the Nifedipine and the omeprazole DDI cases using PBPK modeling approach. The first step was to investigate the drug-drug interaction between omeprazole and the Nifedipine with IR formulation. The simulation was done showing here to mimic four scenarios: A, 10 milligram Nifedipine IR tablets with placebo; B, 10 milligram Nifedipine IR tablets with daily 20 milligram omeprazole pretreatment for 80 days; C, 10 milligram Nifedipine IR tablets with 20 milligram omeprazole pretreatment only for 30 minutes prior Nifedipine intake, and D, 10 milligram Nifedipine IR tablets with daily 20 milligram omeprazole treatment for 80 days. So as you may say, overall, the model described observed data relatively well for the Nifedipine IR formulation. Then what about the Nifedipine extended release formulations? Let's now first take a look at the Nifedipine ER formulation drug release profile, as I also mentioned earlier. So to evaluate the effects of the different pH dissolution media on drug release, a two-stage dissolution study was performed on two Nifedipine ER 60 milligram tablets, one ER product from Pfizer and another product from Pharmaceuticals. During the experimental measurement, one tablet was placed in dissolution vessel containing stage 1 dissolution media, either with pH of 1.2 or with pH of 4.5 for first 2-hour dissolution testing. After the first 2-hour stage 1 dissolution test, the vessel medium was replaced with the dissolution medium of pH 6.8 for the 22 hours stage 2 dissolution testing. The dissolution testing was performed using USP apparatus tool for each formulation at 37 Celsius with a paddle speed at 50 RPM in 900 milliliter dissolution medium under each of the above pH condition for total of 24 hours. So showing on the right panel is the two-stage in vitro dissolution profile at different dissolution medium pHs, where blue colors represent one ER product at two different dissolution medium pH that shows pH-dependent Nifedipine release profile, while the red color represents another ER product at two different dissolution medium pH that shows pretty much the Nifedipine pH-independent release profile. As you may see, almost no Nifedipine was released in the first 2 hours at two different pH values of 1.2 and 4.5 for pH-independent ER tablets shown in red color. While in contrast, you may say the pH-dependent ER tablets release more than 10% after the first 2 hours at both pH condition as shown in blue color. So those in-house data were then used to build the Nifedipine PBPK baseline models for ER formulation. Now back to PBPK model development for the Nifedipine ER formulation. So we had Nifedipine IR PBPK baseline model as we discussed earlier. We also measured the Nifedipine release profiles at different dissolution pHs for two Nifedipine ER formulations, one showing pH-independent and another showing pH-dependent drug release characteristics. What we would like to do next was to incorporate the measured drug release profiles into the IR PBPK model to build the Nifedipine ER PBPK baseline models. So here is a high-level summary of the Nifedipine ER PBPK baseline model. Nifedipine is a biopharmaceutical classification system, or BCS2 drugs with low solubility and high permeability. It is extensively metabolized to highly water-soluble inactive metabolites, primarily by CYP3A4 and CYP3A5 enzymes. The simulations were done using single dose 60 milligram ER tablets with different formulation design, pH-independent release versus pH-dependent release as used in the corresponding clinical studies. Shown here is the simulated drug plasma concentration time profiles in healthy populations following administration of single dose 60 milligram nifedipine ER tablets using nifedipine baseline models. On the left is the result with pH-independent formulation, while on the right side is the result for the pH-dependent formulations. The solid lines are the simulated mean values, dotted lines are the fifths and the 95th percentiles, and the green lines are for different trials. Data points shown here are the clinically observed value that I just showed you earlier. So here is the workflow for using PBPK modeling and simulation to study the DDI between omeprazole and the nifedipine with different ER formulations. First, baseline PBPK nifedipine models were used to predict the PK profile of nifedipine pH-dependent ER tablets shown on the top left panel in blue, and the pH-independent ER tablets shown on top right in blue in healthy populations. That was also discussed in the previous slide. Then DDIs was considered by accounting for the elevated gastric pH shown in the middle panel for both formulations. And then lastly, CYP3A4 enzyme inhibition was also accounted for due to co-administration of multiple doses of omeprazole, as omeprazole and its metabolites inhibits CYP3A4 enzyme, as we just discussed earlier. Shown here is the simulated time courses of the time-dependent inhibition effect of omeprazole on the percentage of active CYP3A4 enzymes in the gut and the liver, respectively, after a daily omeprazole single 40 milligram dose treatment. After 7 days of a daily single omeprazole treatment, the active CYP3A4 enzymes in the gut and the liver were reduced to about 84% and 60% of the original activity, respectively. Shown here is the simulated nifedipine drug plasma concentration time profiles in healthy population after administration of single dose 60 milligram nifedipine ER tablets with pH-independent formulation shown on the left panel, and the pH-dependent shown on the right panel, with daily omeprazole pretreatment for 7 days prior to the nifedipine administration. So here, solid line, black lines is the simulated mean value. Dotted lines are the fifths and the 95th percentiles, and the green lines are for the different trials. The blue lines is the simulated mean value assume a change in CYP3A4 Vmax, which is a maximum rate of metabolite formation by CYP3A4 enzyme of 78%. The data points are the clinical observed values. This table summarizes the simulated and observed nifedipine PK parameters, Cmax, AUC, and also AUCR, which is the AUC ratio between nifedipine with omeprazole pretreatment versus nifedipine without omeprazole pretreatment at different DDI scenarios. As you may see, the change of the pH only did not change the simulated Cmax and AUC that much. So the data shown here suggests that the elevated gastric pH following multiple dose administration of omeprazole has a minimum effect on nifedipine PK, whereas CYP3A4 mediated DDI is likely the main driver to the observed change of nifedipine PKs in the presence of omeprazole. In addition, compared to the pH-independent formulation, as shown in the top part of the table, the slightly increased exposure of nifedipine was accounted for and captured by the enhanced drug release in the pH-dependent formulations shown on the bottom part of the table, which is also captured by the model. So in summary, the PBPK modeling and the simulation suggest that the elevated gastric pH following multiple dose administration of omeprazole may have minimum effect on nifedipine PK for nifedipine with ER formulations, while CYP3A4 enzyme-mediated DDI plays an important role in the clinical observed nifedipine exposure changes. Overall, the effect of nifedipine pH-dependent versus pH-independent ER formulation on the nifedipine and the omeprazole DDIs may be clinically insignificant but statistically significant. So this case study indicated that PBPK modeling approach can provide a mechanistic understanding of the observed DDI of nifedipine ER formulations with omeprazole, an acid-reducing agent. Further case study may be helpful to understand the complicated gastric pH-related DDI, and currently, DQMM with OGD is also conducting research on using similar PBPK modeling approach to predict PPI effect on other IR products. Lastly, I would like to acknowledge my colleagues for all their support for this project. So FDA ORS, Andrew Babskin, Maxim Lee Major, who is now in Simulations Plus, Fang Wu, MJ King, Lu Sifan, Liang Zhao, Lei Zhang, Robert Lamberg, and FDA OPCO Zhongming Gao for performing their in vitro testing. Xinin Yang from FDA OCP, and the Ligong Patel from Certara. So thank you all for your attention. I'm happy to take any questions you may have. And in the meantime, you may also contact me through email if you do not have time to we do not have time to discuss here and answer all your questions. Thank you.
Thank you. That was great. I'll just open the floor to any questions if anyone has any. If anyone has any questions, now's your opportunity. Masood has a question actually, and it relates to the FDA position on accepting PBPK for PPI interactions. What is the FDA position on that?
Thank you for the question, Masood. You mean the specifically PBPK modeling approach to PPI-related study? In that regard, I would say, it really depends on the cases or the models the sponsors are building. I think if it's really like case by case basis, if the model is really well developed and suitably validated, I think that's would be helpful to understand it. For example, for some complicated products with the PPI related DDI, I would say that would be helpful for both sponsors and also regulatory agents to understand the complex mechanism behind that. I think that would be really helpful in terms of the, how much impact that would be on a specific application or cases. I would say maybe it's really a lot easier to say that. It really depends on the model and the purpose and the application of the model for that specific application. I would say we would encourage, this kind of mechanistic understanding to answer some challenge questions in this, very challenged area. Yeah, thank you. I'm not sure if I answered your question, Masood, or not. Thank you.
Yes, that would be great. Thank you very much. I think maybe a follow-up. Sorry, Omaka. What is your thinking about a negative like PPI interaction? If PBPK show that there is no interaction, would that be, yeah?
So you mean if the PBPK models shows the negative and low DDIs caused by the gastric pH change, that's some evidence. If there's the model again, if it's like well validated, I think that's something we can, if that's a real case, if that's a representative of a real situation, that's good. That's good news. We can know, okay, there is no strong DDIs caused by the gastric pH change.
Okay, thank you.
Yeah, thank you.
Thank you. There was a question by Amika, and she was asking whether you've explored using the new semi-mechanistic gastric pH DDI model. I don't know if you're aware of it. So obviously you were looking at kind of static pH, but there is a model which is implemented that can look at dynamic changes in pH.
Oh, you mean dynamic changes in pH for the DDI? So I'm not certain if I completely understand the question, but I think you are talking about the like in our model here, we used like the dissolution, data from two-stage dissolution data. One at like normal, pretty much normal gastric gastric pH or 1.4 or 5, and another one is elevated gastric pH. We have only incorporated 2. I'm not sure if you refer to that or not, but I think that's a good point. So currently, for this model, we only use like either or, like the pH without the pH elevation versus pH elevation. So if there is a dynamic model to mimic that, that would be great, I think. But currently, we based on the in vitro dissolution data, we incorporated that into the model only, not the sort of static. I agree with the not dynamic model for that part. However, I think in the model itself, I mean, the pH change, during the GI tract, for example, gastric, and also different GI tract has different pH that may also affect the result. I mean, by the simulator, it count for that part. For example, for the biorelevant solubility at different GI tract segments, I think that's sort of counted for, but back to the basic profile used, yes, we just use 2, in vitro measured profiles with 2 specific pH. That's static. I assume you mean that part. Thank you. I'm not sure if I answered your question, but yeah.
Yeah. Thank you. Yeah, you did. And in fact, there is a technical webinar that covered this earlier in the year, if that's interest to yourself or anyone else on the line. If there are no more questions, I'd just like to. Oh, sorry, there's one more question. If there had been any cases the FDA has accepted PBPK modeling for a PPI instead of running the clinical study?
That's a good question. Because I'm not in the new drug side, I'm on the generic drug side. For generic side, I don't think so. But for the new drug side, I'm not aware of that as well. Maybe I'm wrong. I could get back to you to confirm that with my new drug colleagues. But as I know, I'm not sure. Actually, probably not. That's my best guess. You mean solely based on the PBPK modeling approach without do any experimental data for some product? Yeah, I'm not exactly sure for that question. I'm sorry about that.
Okay. That's fine. Yeah. Okay. Thank you again, Ming-Mei, for your time and for presenting to us today. We don't have any more webinars lined up this year, but watch this space for the webinars that are going to be in place for next year. So just like to thank everyone for their attendance and see you in the new year.
Yeah, thank you all. Thank you, Oliver.
Thank you. Take care.
Thank you. Bye.
Bye-bye.